Welcome to the Masters of Beautiful Achievements podcast, where we share the insights from inspiring masters about how the use of physics, biology, and green chemistry is shaping our future. It's all you need to know about innovation and better business models. The podcast is hosted by Alexander Prince, founder of Scope Matters. For more inspiring episodes, visit the Masters of Beautiful Achievements website. Today's episode is part of a special series where we look at microbes through a different lens. We will explore how life sciences, biotechnology, and environmental sciences are influencing how we look at our natural world. During this series, Alexander Princeton and Mark van Bemmel are curious to know what happens when we zoom out and look at microbes as important players in a much broader system. How does this different perspective influences on how we look at the role of microbes and viruses in the web of life? And what lessons can we draw from nature's logic into making our world more sustainable? Together with specialists in the field of virology, microbiology, and biochemistry, we go on a journey combining insights from various disciplines to learn about the essential questions we might have been overlooking. And who knows, we might discover one or two innovative applications along the way. This series is brought to you by Scope Matters, a systemic innovation agency from Rotterdam, the Netherlands. They accelerate business leaders through systemic innovation by merging ecological thinking with economic doing. Their system approach transforms and improves the business model of their clients by creating new opportunities, mitigating risks, and increasing cash flows, while at the same time eliminating waste and doing good. It is the space where systemic sustainable thinking meets circular innovation, because in the end all matter counts. Check out the website on www.scopematters.com. And Orvion, a young biotech company based in Stolwijk, the Netherlands. They study microorganisms and are developing their own DNA analyzing techniques, like for instance the Orvion U-Detect. They work on solutions to remove contaminants from ground, water or industrial processes more cheaply and permanently. Their services are available for water treatment, soil remediation, ecology and industry. Orvion believes nature can do a much better job, so please take a look on their website on www.orvion.nl. In this episode, we will investigate how it is possible that even bacteria can produce a type of bioplastics, which is actually called PHA. And even more remarkable, this PHA turns out to be also a natural feedstock for them. Why and how do they do that? And what can we learn from microbes about their, and even more interesting, our own metabolism? Today we invited Gert Jan Euverink from Rijksuniversity Groningen. Please, Gert Jan, introduce yourself to us.
Yeah, hello, my name is indeed Gert Jan Euverink. I'm a professor in biotechnology at the University of Groningen. I'm heading a research group where I train PhD students in the area of microbiology, biotechnology, and hopefully to contribute to a sustainable society and a circular economy. So I combine science and business in my research. I try to design processes which needs to be picked up by companies to make it bigger. So the things I do is usually on lab scale, laboratory scale. So 1 liter, so kind of the volumes you are accustomed to as what you do in your kitchen. And sometimes we go to a little bit higher volumes, but at the most 1,000 liters.
Okay, thank you.
Microbiology is the central point in my research.
Mark.
Yeah. I'm Mark van Bemmel. I'm CEO and founder of Orvion. Orvion is a company dedicated to making the world a bit cleaner by using microbes. So we are really focused on finding microbes that can degrade all kind of harmful substances. And we use our laboratory, we use DNA techniques to show if they are present and in what numbers. Also, we do a lot of work on finding technologies to use the microbes in a positive way, so that we can produce chemical substances in a friendly manner, because microbes are very effective. They use less energy. You don't need a high pressure or a high temperature for them to work. So there's a lot to gain if you use microbes for doing this kind of stuff.
Thank you, Mark.
Sort of the headline, yeah.
Yeah, and a good start of today.
Yeah.
Because we have Gert Jan Euverink in the call of in this session, we're going to start off with bioplastics from bacteria. He's one of the experts in the Netherlands working with PHA, I assume, and PHB, I assume. But you can explain that later. So to start off, the how, what, and why of this future material, Gert Jan, is it promising and why?
Well, the material is promising because it is, first of all, it's biodegradable. So if it ends up in the nature, the microorganisms in nature will degrade this, and there will be no remains in the environment after the bacteria have been growing on these kind of substances. So the problem with the microplastics or plastics, what we have now in the oceans and in our environment, it does not occur when you use bioplastic or this PHA.
And for those who do not know PHA,
that's polyhydroxy archanoids, and this is a kind of a plastic material with similar properties as plastic made from oil.
And it grows in the bacteria itself.
Yes, this kind of plastics is made by the bacteria. So you grow a lot of bacteria, and then you apply conditions where the bacteria will fill themselves almost to 90% of their internal volume with this kind of plastics. And then we have to get it out, and then we can use traditional plastic equipment to actually make thin films out of this or bigger plastics.
Mark?
Yeah. Gert Jan, do you know why these bacteria do this?
Well, these bacteria don't make plastic, of course. They have no use for plastic. So what they do is to use this as a kind of a storage material when there is a need for food and they are coming into an environment where there's no food. They still have something. This is like similar as from the potato. The potato has starch in the roots, and they are used to grow a new plant. So under the new conditions, they will use this material again. And the bacteria will do actually the same. If the conditions are not right for them to multiply as fast as they want, and they still find only part of the food, they will store this as kind of bioplastics inside the cells. So it's
and they carry it with them all the time?
No, not all the time. It's depending on the conditions. And bacteria, they need carbon and they need nitrogen to grow. Yeah, just like a plant, like they have fertilizers. You need carbon, you need nitrogen, you need phosphate. But in those cases where you have no nitrogen, but still have a lot of carbon, then they are going to make this kind of reserve material. And when the conditions are as such that there is sufficient nitrogen available, then they will eat up this material, and then they can start growing again.
What kind of sources do you need then as nitrogen and phosphate?
The nitrogen is coming from ammonia.
Okay.
Is that what you mean? So where does it come from? Yeah.
What is the grow medium, and where do you get that from?
Well, in this, the growing media is carbon. So you can have acetate, or you can have glucose, or you can have glycerol. So all kinds of substances.
They will grow on acetate.
The bacteria will grow on acetate.
Oh. That's whenever you say, of course, but I also know that you have certain bacteria that actually produce acetate.
Yeah, indeed. So that's why you have lots of different bacteria in the environment, and they have kind of equilibrium. So some of them produce acetate, but some of them are also able to eat acetate. So if this was not possible, then our Earth would be full of acetate. If it's not consumed by other bacteria, then over millions of years, you would have an accumulation of acetate.
Would be very acid.
Would be very acid, indeed.
Yeah.
Then we have lots of vinegar.
Yeah, but then acetate is also a drop-in chemical for the petrochemical industry, and to make plastics.
Yeah, indeed. So this is also one of the ideas that you have bacteria that can make these polyhydroxy archanoids, these bioplastics. But you also have different types of bacteria who can produce acetate or who can produce lactate. And from those substances, the chemical industry can also make bioplastic. It's a different type of plastic. These are not made by the bacteria. The individual components of the plastic are made by the bacteria. And then the chemistry glues all these molecules together, and then they make a large, big molecule, and that's also called bioplastic.
Gert Jan, do they only use acetate and that kind of glucose, or also CO2, for instance?
There are some bacteria that can also grow on CO2, but those bacteria, they need actually light to grow, because carbon dioxide has no energy. There's no energy value in there. It's an end product.
Right.
That's why we have this problem in our environment. So they need energy. And some bacteria, they use light for this, just in the same way as a plant. But you also have bacteria that use hydrogen as an energy source. And carbon dioxide and hydrogen is a combination that some bacteria can grow, and they can make biomass, they can make proteins, and proteins is part of our food. So if you do this right with the right bacteria, we can make food from carbon dioxide and hydrogen.
But then Gert Jan, you're mentioning, like, I mean, if you have a protein, you can also make polymers.
A protein is a polymer.
Yeah, so you have the PHA, which is a sort of a membrane energy storage. They can produce protein, which you can make of plastics. They can produce acetate, which you can make plastics. You're basically saying that the bacteria are the future plastic powerhouse, as long as we feed them the right stuff.
Yeah, but not everything is done in a single bacteria. These are all properties from different bacteria, and so you cannot make this as a single process. You have to have separate processes. There's a process that makes bioplastics that cost energy. There's a process that makes proteins that you can turn into food if you want. There's a process for bacteria that make acetate or lactate. And all these different processes, then you need chemistry, or you need biology, engineering, to make actually the processes and the products you want.
So what are the challenges right now, Gert Jan, to make PAH on a larger scale? What are you trying right now to get it done?
Yeah, what we are trying is to get the bioplastic, the plastic in a suitable purity that the polymer chemists can work with it.
And they are so critical people, isn't it?
We have to compete with the oil industry, and usually competition is done on the basis of price, not so much on the basis of properties. And because everybody knows polypropylene has nice properties, and they don't consider something else. And so everything is based on price. So if we want to make packaging material which should replace polypropylene with these bioplastics, then we need to be cheaper than the polypropylene.
Or better.
Yes, indeed, indeed, that's better. That's also my philosophy. We cannot compete on price at the moment. A barrel of oil is something like $30. I don't know the exact prices, but it should be somewhere in that area. A couple of years ago, when there was a boost in this kind of technology, the barrel of oil was $120. So there we were able to compete a little bit on price. But I think that we should not compete on price, we should compete on properties. So we should be able to make with bacteria bioplastics which performs better for the products they are used in. And we already have an additional gain, is that our material is biodegradable.
And what is biodegradable? Because that has so many definitions at the moment.
Biodegradable, in my opinion, is that it's completely converted into biomass, bacteria, and carbon dioxide. So that there are no remains in the environment.
So if you could use actually PHA if you make plastics, they will start adding additives into that to improve the quality of plastics. In this case, PHA. So you have PHA, which could be polluted with other additives.
Yeah, that's true. So we also have to be careful on what kind of material we add into this biopolymer. And what actually maybe we can make by the biopolymers with the bacteria that do not need this material.
Mm-hmm. Okay, but that's always the discussion with plastics and people when they're saying it's biodegradable. Yeah, that always depends. Somebody gave me a definition of bioplastics, and if it contains minimal 20% or something, or 30% plastics derived from a biological source,
yeah,
you already can call it bioplastic.
yeah, okay,
And
yeah,
the remaining 70% is fossil, and then you have the discussion. Okay, but how can it degrade? And degradation of plastics is always an issue, because plastic was created because it was a very good packaging material.
yeah.
And if you have packaging material which dissolves within 24 hours, you have another issue.
Indeed. So it's you cannot use this for all applications. But if we could decrease the amount of plastics that is used, even for short-term purposes, that would be enormous. Because the short-term purposes, they end up in the environment. Eh? People throw away plastic bags and so on. But if you want to store something longer, you have more. Then the product might not easily end up in the environment because it's longer on the shelf.
Mark?
Yeah, we have a question from Ineke. She asks, do you use natural bacteria or genetically modified?
CRISPR.
CRISPR-Cas as well. Now, for the processes we develop at the moment, because we are working with biomass and waste material, we use natural occurring consortia. We don't use genetic modified organisms for this.
Do you believe anyway in modifying bacteria to get better in this, or is it? Do you have another opinion on that?
Yeah, no, I think certainly it's an option to use genetic modification, but these organisms are usually not as strong as the natural occurring organisms in the environment, and so you need to sterilize all your starting material. So I was just mentioning, competing on price, treating your starting material is expensive. Yeah, you need to sterilize, and that means heating to 120 degrees Celsius.
Yeah.
That takes a lot of energy, and that makes your process even more expensive.
And that's what you do not want when you have a biological process. You want to do it at current normal temperatures and normal pressures and so on, because that's the value of this process.
We are trying to create. Sorry.
How does LanzaTech do that? Because they started off with a methane-eating bacteria from the rapid gut system.
Yeah.
And they have pushed it not genetically but through evolution. The survival instinct to create 50 acids and fuel components.
Yeah. Then they have with evolution that you do then you use the natural occurrence of mistakes in the DNA, and then you select every time the proper bacteria, so the ones that produce a little bit more than his parents or her parents, whatever you want to say.
Yeah, because in their let's say iteration process, they discovered that the bacteria they were using had actually 3 different metabolistic routes, depending on where you how you starve them and how you feed them. They would choose one of these 3 routes, and then as a result have an explosion of acids and butanediol. I don't know if that is actually an acid, but it produces chemicals ultimately as an output, because it was sort of it had to find a way to run its own metabolic system.
Yeah. No, indeed, you can do this, and if they're growing on methane, that methane itself is a gas and it's already sterile, so you do not have to sterilize your material. But still, it has a certain value. And what we are aiming for is to actually use the wastewater and the carbon, which are the carbon molecules which are in the wastewater, as a source for making bioplastics. So we are doing 2 things at the same time. We are treating our wastewater, and the bacteria will produce for us the amount of the bioplastics, the PHAs.
There was one more question, Gert Jan, from Jules Rombas. Welcome, Jules. Is it possible to make transparent PAH granules as end products from more waste type feedstocks, let's say food waste?
I don't know enough about the transparency of PHA, but if the PHA molecule itself is transparent, then the food and the starting material doesn't matter. They all degrade the food and the carbon until polyhydroxy butyric acid or polyhydroxy valeric acid, and then they synthesize the polymer. So everything is converted into something similar, either proteins or glycerol or ethanol or whatever. The bacteria will do this, and there's a common molecule, and from that common molecule, they start to make the PHA.
Ah, okay. And do you need different species of bacteria that start from different molecules, or is it all done by one or a few strains?
Yeah, it is a bit difficult. It's difficult to say. There are a lot of bacteria which are very versatile, so they can handle a lot of different foods. But you also have bacteria that can handle only a single type of food.
Yeah, highly specialized. Yes.
They're very specialized, and so depending on the environment where they were growing originally, if it's necessary to survive, if you are very versatile, so then you have different kind of food available at different times, then you need to adapt. But if your environment is always the same, then you can really specialize, and then you can become the winner of such a system, because you can do the process and you can use the metabolites at the highest possible level.
Yeah, because you could expect that there would be a lot of competition for things like acetate. I mean, that's so easy to degrade for many bacteria.
Yeah, but there are many bacteria, and they are already there for a very long time. So it is not something that you can easily reproduce in the lab if you want to go to stable ecosystems like you can find in nature. Mm-hmm. But we do our best.
How are you succeeding? For instance, if you use acetate, is it like 80% that ends up as PAH or 10% or?
We have not done yet the mass balances.
Sorry.
But the cultures that are using the PHA in our communities are 60-70% PHA producing organisms, because we apply conditions that only the ones that have storage material in them survive. And so we are mimicking nature. So in cases where you have in the environment maybe a lot of rain, so then all your material will be flushed away, and then the bacteria have no food. So in essence, we do something similar. So we are removing the food, and only keep these cultures without food for a while, and then the ones that have no storage material cannot grow and cannot survive, and the other ones do. And then we will start again. So it's a cycle we apply, and so we are making the ones a winner who produces storage material.
And I have two questions. One of them is that you are suggesting that you actually are working with mixed culture systems.
Yeah.
And that is very new in the fermentation or area, because everybody's used to do monoculture also in bacteria. And
I disagree with you on this.
Oh, okay, but at least I know a lot of actually two things. One of them, Jules, who was not here. I know him from the TU Delft.
Yeah,
he's from the group of Mark van Loosdrecht, and his PhD was actually around mixed culture processes, which was quite new to work with. Most people are used to just to have one species and then work with that.
Yeah.
And actually, and you're saying is that multi-mixed has actually even better outcomes than single.
All our wastewater treatment processes are mixed cultures.
Ah, okay.
So we have a lot of experience handling mixed cultures because our wastewater is treated every day. So we can really deal with mixed cultures. Another example of mixed cultures is making cheese, making yogurt. This is done by mixed cultures, and they are controlled.
Interesting.
So only the ones who want to put bacteria in a reactor in an industrialized environment, they would like to have full control.
Yeah.
And also because they only know that a single bacteria, they can do this kind of evolution. And that is not usually better for the bacteria to win a competition. If we do evolution and that the bacteria will produce more, it will have less energy available for growth because the energy goes into the products.
Yeah, that's
and
also
that
a fact.
means that we have a single bacteria. So that's why we want to have single bacteria in industrialized processes to make, for instance, antibiotics. The penicillin process is done by a fungus, and that fungus has been changed over years by natural selection and natural mutations by Gisbocaris. And that organism cannot compete with the parent organisms. So you need to have only this organism in your reactor, or the other organisms will win.
You don't want to put the weak laboratory guys into use.
No.
And actually, what I've learned about PHA, it is actually the first steps of a biodigester, which is still amazing for me. So the question is actually, how did they discover PHA?
How did they discover PHA? I think in my university, they were growing organisms on oleic acid, and this is done in the 1990s. Yeah. By the late Professor Bernard Withold in Groningen. And when they were growing these organisms on oleic acids, they saw huge some organelle-like or vesicles-like structures inside the cell under the microscope. But I'm not sure, he did not invent this. This was way before known. And I don't remember the first literature where they measured PHA for the first time.
But it's a very recent finding, so it's something from 20, 30 years ago.
Yeah.
And how is it connected with the biodigester? Because I've learned that the first two steps in a biodigester is actually the PHA production.
Yeah, in a biodigester you make methane, but acetic acid and organic acids are the intermediates.
Okay.
So when the biodigester grows on biomass or on proteins or on fats and so on, they all convert this into organic acids, and the organic acids are converted into acetic acid, and then they make methane. So if you are able to change the conditions that the whole community stops at the production of organic acids, you have more of these organic acids available, and then other organisms can start growing. And some of them are indeed the organisms that make the PHA molecules.
Gert Jan, I guess that the PAH bacteria, that they need oxygen, is that right?
The ones that we have at the moment, they need oxygen. They grow oxygen, yeah.
Yeah, so they're aerobic.
So but Gert Jan is suggesting also that you don't need oxygen. Some don't need oxygen, or?
I've seen in literature, but I don't recall exactly the names of the bacteria, that also anaerobic organisms can accumulate PHA.
Ah, okay. And do you have the impression that it is quite a regular feature of bacteria, or is it really just a few bacteria that can do this PAH storage?
Oh, now you're asking me something. I did not do the genetic analysis yet, but I think there are quite some clusters present in bacteria that can actually make these kind of storage material.
So,
but it's not that common. It's not that you isolate a bacteria that you will have 90% chance that this will be a product.
Okay.
So the other reserve material bacteria use is glycogen. So that's a polysaccharide. Yeah. It has the similar property, has the similar idea, the function as the PHA for bacteria, but then it's glycogen. And the same do animals. We as well. We store glycogen as storage material, and plants have starch.
Yes, it's like a sugar type.
Yeah, it's just sugar types.
Okay.
Yeah, but that also means that brings me to a question more from a practical point of view. Let's say I produce a lot of milk. I'm a milk dairy farmer, so I have a lot of manure left.
Mm-hmm.
Actually, you're suggesting that he has a choice to make bioplastics or methane.
It's a bit more difficult with manure because the amount of nitrogen in there is preventing that the bacteria will feel a nitrogen limitation and that they can start producing the PHA, because they do this when you have no nitrogen and still have a lot of carbon.
So you have to, so you have to remove the nitrogen from the manure, in fact.
Yeah, and if you know this, then you have then the Minister of Agriculture will be very happy with you.
Yeah.
Because that's our biggest problem at the moment. The ammonia. So if we can solve that, it would be nice.
Yeah, although I mean, another route which I had during my Dutch podcast series, the New Four, was a guy from Bij de Oorsprong who works with EM organisms, effective microorganisms.
Yeah.
The Bokashi method, and he actually, what is it English word for rijpen? I don't know if there is an actually word for
ripening.
Ripening.
Yeah, for ripening manure. And then it becomes from sour, becomes sweet, and then it actually is good enough to put back on the land. And then there suddenly nitrogen is a good feedstock.
Yeah, we do need a certain amount of nitrogen as fertilizer. So if the cows eat the grass and the manure is treated at other places, then eventually you will lose the nitrogen in the soil. So you should be able to bring back the nitrogen back to the soil, but not as much as we do at the moment. Yeah, because we give the cows additional food. Yeah, we give them additional soy protein, so the amount of nitrogen is a surplus, because we want to produce a lot of milk and a lot of meat. And if you do not supply the cows with sufficient amount of nitrogen, they are not making as much milk and not as much. They don't go as good as their meat producers want.
So if you would have a bacteria which eats nitrogen, what kind of metabolic reaction will you get?
You will have the reaction from ammonia and nitrate to nitrogen, and then it will go into the air. But you can make a harmless product from ammonia.
Yeah, but you can also capture, of course, the gas and then bring it to Air Liquide or another company.
Yes, but it's a little bit useless because the nitrogen is already in the air. We have 80% of our atmosphere is nitrogen.
Yeah, but if you create a bioreactor where you throw in manure and then the eating nitrogen bacteria, then you have nitrogen as an emission.
Yeah. But there's no use of capturing it and bringing it to Air Liquide, because in the atmosphere is that much. They can much more cheaper get it out of the air from their own environment. But if you
but then we talk about geoengineering, and then
no. The amount of nitrogen which goes into the air has absolutely no influence on the atmospheric quality.
Yeah, but then we also have the discussion about the CO2 emissions from agricultural systems, which
yes,
it's a natural carbon cycle, so that is also a bit of
but the problem is that even 90% of nitrogen does not change our climate. We have a problem with the amount of oxygen for us, but nitrogen itself, then you have only 10% oxygen. No, but the nitrogen itself does not contribute to global warming, and the amount of nitrogen remains the same. And what we did with the oil is that our storage material of the oil is coming from decayed trees and biomass, and that is stored over millions of years in the earth. So, and the plants, they grow on carbon dioxide. So they have been extracting carbon dioxide from the atmosphere, and now everything is now put into the soil. But the amount of material which has been collected over millions of years is now being burned in just 100 years. And that is the reason why the carbon dioxide concentration increases, because it's collected over millions of years and put into the environment in just 100 years.
Yeah,
If we also would use all these millions of years on storage, and that we are using them as careful as possible and with low amounts, then there would be no problem, because then it's just part of the natural carbon dioxide cycle.
So we should work on the uptake part. So take up a lot more CO2.
Yeah, that's why we are promoting natural environments and forests, and that all these organisms are still there to capture our carbon dioxide.
Right. And revitalizing soils is extremely important, in my opinion, to restore the natural uptake of CO2 also.
But also means that you need more natural bacteria back into the soil, because the soil flora has also been degraded.
Yeah, sure. And so if you would find a way to reforest Sahara, for instance, that would really make a difference. But okay, it's a bit off topic now, I'm afraid.
I think also Jules has another profound question. What do we think about producing PHA with nitrate and nitrite as an electron acceptor instead of oxygen? Oxidizing ammonia from manure and then converting them into nitrogen gas and PHA.
Yeah, could be a nice solution if we are able to do that. Then it's just you just have to find the organisms who can actually work in these environments. And we have a very good professor in the Netherlands who is looking for all kinds of, almost he says, impossible organism. And that's Professor Mike Jetten from Nijmegen. So we have to talk to him then to find the right conditions to do this. And maybe he already thinks, he says, oh, well, this is done. You have to use this organism or that organism.
Just from a bioplastic perspective, how does PHA, how can it be compared to PLA?
PLA is made in a chemical process. It is not that easily biodegradable.
Yeah.
And the monomers, so the components where they make the PAH from, is made in an industrial process by bacteria.
Yeah, it's fermentation from starch and from mice, I think.
Yeah, from lactic acid bacteria they produce. Corbion is in the Netherlands the company who makes lactic acid, and they are also involved in the bioplastic production.
But then I'm also then curious, because if you have kombucha or Scooby, let's use that word, the Scooby, which you make kombucha, it also creates a biofilm on top of the medium, and some people, I think it was also in Rotterdam, are making fruit leather from that. Laurie Golf is making a type of bioplastic from it.
Yeah.
Is that also PHA, or are also microorganisms able to produce a sort of like a biofilm polymer?
I think if you talk about the leather process, there are fungi doing this, and from the fungi itself, they turn this into a kind of a leather material. So it's not a product they make; they use the organism itself.
Okay, and do you know about microorganisms that actually produce a biofilm as an output?
Many bacteria produce biofilms, and at many locations. So if you put your finger in a water tube, the slimy material you find is a biofilm.
But that's also a polymer.
That's a polymer. These are usually polysaccharides.
And can you convert them also into a type of bioplastic? Because if it's a natural output.
Yeah, maybe, because you can also convert starch into a type of bioplastic.
So but then Mark, we should find a guest speaker who actually knows something about that, because then it's a big problem in a lot of industries. But actually, if you turn that problem into an opportunity, then those biofilms become a bioplastic, let's say, feedstock.
Yeah, but I think that we just mentioned that PHA is biodegradable, but it's not extremely fast. So it withstands a certain amount of time in the environment. But the biofilms from polysaccharides are quite easily degraded. So you lose them very quickly.
It's
then
not,
it depends.
not stable, you say, Gert Jan.
Yeah.
And then it depends on the application for that.
Yeah. That's true for most material.
I guess also the problems in industry from EPS, that's how it's called, extra polymers substances.
Yeah.
Quantitatively, they are probably, you're talking about very small amounts, I think, generally, but they can cause problems by clogging
it's going to be a challenge.
Pump or whatever.
Yeah, it's going to be a challenge to produce a lot of them.
Yeah.
But then the challenge, I know Laurie Gall from Unplastic from Blue City in Rotterdam. She is working in that direction and actually has a product that actually has properties which can be applied to packaging. And I think her next step is upscaling that process to enough volume that you can actually attack the market from that perspective.
Yeah.
You already touched the point, Gert Jan, that we have to get something that is, that has better properties, or at least as good as other commercial fossil plastics. But is it also possible to mix PAH with other fossil plastics? Like we have the E10 now in the gasoline at the stations. Yeah, we mix in some biological produced ethanol, for instance. Can you also mix in PAH just to get it rolling, to also get some scale for the PAH market?
Yeah, you can of course always try to blend the material if they have if the mixture is stable. So it depends a little bit on the type of polymer. So suppose you have a polymer which is able to melt. Yeah, it becomes soft when it's hot, and it becomes tough material when it's hard. So
Right.
if you are able to mix in different kinds of polymers, and then the final product would be better than the single products from the beginning, yeah, why not? But then it's going to be, you still have material left after the plastic ends up in the environment. And maybe they are even worse, because if bacteria remove the biodegradable parts from this mixture of plastic, you will have a degraded, half degraded material of the other plastics. Right. And then they fall apart. And maybe then the amount of microplastics, which we now see in all the places where we even try to measure something, would even be worse. So then you make a product which is maybe nice, but has, potentially...
The worst properties on the long term, yeah.
Yeah.
Gert Jan, you mentioned the biodegradable of PHA. How does it degrade? Because you mentioned it's a bit stiff, so it takes time. So it needs some time to break open. Let's put it that way.
Yeah.
Can you share with us how it actually breaks down?
So the bacteria needs usually water to break down material and also to grow. So water vapor needs to enter the material, and that goes quite slowly. And with the water vapor, enzymes will be brought into the material, and then they degrade the material from the inside out. It's a process which is comparable with the degradation of wood. Yeah, wood is also quite resistant. If you have a tree in the forest, it will take some years before it's gone. And this is also a slow process. So enzymes from the outside need to go to the very small pores inside these plastics, and then they degrade this.
But
it's possible, of course, because the bacteria who produce PHA are also able to degrade it, because they use this as storage material. So they need to make it, and they also need to break it.
Right.
Then it remains the question: what is the basis of the enzyme? Is it a bacteria? Is it a fungi? Is it the yeast, or even animal-based?
Could be most, if I have to guess, maybe fungi, because the bacteria do not produce these enzymes to the outside, because the Polyhydroxy Archanoids is in the inside. So there's no use for them putting the enzymes outside to degrade something which is not there. Yeah, they need the enzymes in the inside.
Inside the cell, yeah. There are two questions.
Yeah, and PHA are much more specialized in producing enzymes for the outside world and degrade all kinds of material for us.
But then I'm curious. I've seen it, Mark. But are viruses able to then break down PHA?
No.
No, because but then if you use the analogy of a tree, and the trees. I spoke to a scientist, and he said to me, or she said to me, that the moment fungi evolved, the trees were able to degrade, and since then we don't have any fossil fuels anymore. Since that period of the existence of fungi, all the trees that existed before fungi could not have been broken down, and such are the fossil fuel sources we have now.
Yeah.
And now you're mentioning with PHA, that is extreme. I mean, there should be an organism which. I mean, if you eat a bacteria, somebody needs to eat the bacteria, and when you eat a bacteria, you will encounter the PHA.
Yeah.
And if it's a sugar source, the same as starch or glycerine that humans have, somebody enjoys feasting on it. And I'm extremely curious what kind of organism would just be waiting to get his hands on PHA.
Yeah.
But nobody has the question yet. So, but that's my way of thinking. We have Omid has a question of a practical use of bioplastics as a filament or 3D printing on the PHA.
The polylactic acid is already used as a filament in 3D printing.
Can you use PHA for that, or PHP?
Oh, I don't remember. I think the PHA has a bit higher temperature to melt. And that is somewhat of a problem with the current 3D printers. But definitely the material, it can be melted. You can put it through an extruder. So if you have the technical abilities, you can also 3D print with PHA.
And Jules had another question. What would you like to have pH replace as a customer product?
In the ideal world.
Yeah, but I don't know if there is an ideal world. I mean,
a philosophical question.
The change goes so fast that I had a conversation last, and somebody thought we are looking for polymers, and the question is, okay, do you want them from bacteria? Do you want them from fungi? Do you want them from algae? Do you want them from plants or from animals? What do you want? We all talked also that from protein you can make polymers.
Yeah.
We can make from lactic acid polymers, or from organic acid. So polymers are everywhere.
Indeed. And depending on the chemistry, the monomers are usually sometimes not even that of a big problem, because the bacteria can also eat the monomers, also from the monomers from polypropylene. But it's the problem after you make this polymer that they repel water. And if there's no water, there's no biodegradation. So there is a problem. And what material should I exchange with biopolymers? I think I'm going to make a kind of a general remark, is that all the material which are difficult to collect and recirculate. So that's where you have the potential that it ends up in the environment and pollutes our environment.
Yeah, or if you have a plastic bag and it ends up in the ocean, and the ocean is full of water, and hopefully the salt is not an issue, then the bag becomes a feedstock for the ocean bacteria.
Yeah.
We can feed the ocean from our plastic bags.
Yeah, but now you're touching up something that is also sometimes problematic. If you say that your product is biodegradable, then people are not that careful anymore and just say throw it in the environment, because it will break down anyway.
If there is enough water.
But if you have too much plastic inside a system, and then a lot of bacteria will grow, and then it will pollute. The bacteria will make the water turbulent.
Yeah, or oxygen depletion could be a problem.
the same what we have with the blue algae intoxicating. But yeah, but then again, it's the same with, I don't know, if you're dead animal, all the flies go there and all the bacteria and the fungi go there just to get rid of it. But then it all depends on concentration.
But first of all, it's an attitude change. So people should not throw away this material. I know, but after that, if it cannot be prevented, that all the limited amounts of this material ends up in the environment, and if it's biodegradable, you will get rid of it. But it should not be the only solution that it's for biodegradation. Or to prevent that people are not putting this in waste bins or collect this and treat this in a proper way.
Yeah, or you make them responsible for degrading it themselves in their garden.
But
in the composting.
If you take this line of thought, then actually, if you would be a farmer and you're looking for fertilizer, you can collect the PHA bags.
Yeah, but that you don't need carbon as a fertilizer. You need nitrogen and phosphate, and that's not in there. But it doesn't help.
If you look in our garden, you see three big clicos, three big trays where you can separate waste. But they're so huge, you throw in everything. So it doesn't really work in that way, because you should actually make very small ones so that you are more, you think better of what you throw away. So if you would have plastics that biodegrade easily, you just compose them in your own garden.
Yeah, but you can do that with PHA.
Yeah, I think so.
So, but what is PHA? It has no nitrate, so it has carbon, hydrogen, oxygen.
It has carbon, hydrogen, and oxygen.
That's it.
That's it.
Only the three.
Yeah.
Oh, okay, interesting.
And for growth, you need nitrogen, and you need phosphate, and a couple of other small things. But that's the main constituents of living organisms.
Yeah, and in PLA you have the same three.
The same.
Mm-hmm.
Ah, okay.
Just lactate, but then
if you have nylon, there is a nitrogen in there and carbon. But I don't know any polymers which has phosphates.
Fluor they do.
Yeah, but fluor is not it's not very good for organisms.
The PFAS, we had a discussion last time.
Yeah.
That's horrific stuff. Yeah.
But you thought last time, Mark, that there are microorganisms who actually use it as a feedstock.
Yeah, of course, but they are the highly specialized.
But the interesting is that there are organisms actually capable of doing the job.
That's very fascinating, of course, and that's.
If you expand this to what Gert Jan said, you're doing in a lab, and if you want to push it into an industrial system, I'm getting the image of multicultural reactors, and you throw in stuff in one, and you throw in stuff in the second one, and in the end you get new material from it, and then you just put it back into the chemistry or the extruding industry, and then everything is solved. And at home, or I don't know, in a local area or in the city, you have those, instead of bringing these things at a separating unit, they have like 20 or 30 different bioreactors depending on the stuff you throw in.
Maybe, yeah. Is that what you would like, Gert Jan, or?
Well, I'd like to have more reactors which are which you can feed in different types of substrates, because the community is sufficiently stable that they can handle all kinds of mixtures. If you need to have separate reactors, you need a lot of steel and a lot of energy and a lot of people to maintain them. So it's much more easier to have a single big reactor where you can just throw in quite some material. But you do need to know the characteristics of the community, of course, because if you put too much in there which is toxic, then you kill the whole community. So you should be very careful adding your amounts of substrate into these reactors. And this is also true for the biodigesters. Sometimes people throw in a lot of starch or a lot of maize or potato waste.
Yeah.
And then the organisms eating the starch are growing very fast, but they don't make the methane at the end. They only make a intermediate like these acids. And then you see that the pH is dropping in these methane or biodigesters, and then the whole culture is killed. So you need to be very careful. And these are living organisms. The same as if you have a cow or a dog, you don't feed them too much because they will die. And microorganisms also can die if you feed them too much.
Or your goldfish, yes, yeah.
Yeah, but
Or a goldfish.
it's a science in itself. Then, if you run a biodigester, you actually need to know your nutrient content, what you're throwing in.
Yeah.
I always had the impression you can throw everything in a digester and poof, we have methane, which powers everything.
Yeah. Unfortunately, these are quite sensitive organisms who make the methane. They are quite old in the evolutionary line and so on, but they are not that strong and they cannot withstand oxygen at all. Then they will die as well. And they cannot survive under very acidic conditions.
Yeah, and they grow slowly, so you must be careful with them. Yeah.
Lots of ammonia is also a problem for them.
But then, what defines a good biodigester as a service provider? Because a lot of people are offering biodigester as a solution.
Yeah, but maybe you should have two systems in this. That you have two reactors. One reactor is producing the acids, and that can be done under very tough conditions. And then you can slowly feed in the liquid, which contains all the acids which are needed for making methane. And you can control the addition of these acids to these methanogenic organisms and producing the methane. So then you might, if you are working with all kinds of different feedstocks, that would be a solution. But if you are working with a single feedstock, and you can just adapt your process and your size of your reactor and the sizes of your pumps to maintain a single stable reactor. But then you are not extremely flexible. And if you want to be flexible, then you need to have a bit of two-step process.
Yeah, then you also produce interesting acids for another industry. So actually, methane is then is one of the products you can actually produce.
Yeah, but acetic acid is also a nice product for the industry.
Yeah, that's why. So the one, but then it's interesting because especially in the let's say manure. Animal manure, everybody's pushing for the methane production, but actually the 2 steps forward, the organic acid production might actually be more useful than the methane.
Yes, but the amount of acetic acid that the chemical industry needs is huge, and to get acetic acid out of water is not that easy. Also takes some energy. And so for the farmer, it's much easier to use the methane to either as a source for heating or a source for electricity, which he can use right away. Instead of trying to get the acetate out, the liquid, and transport all the liquids, because then you also need a lot of energy. So transportation is expensive.
Yeah, but the future will be that electrons will almost be free. So the transport in itself, the transport cost will go to zero. It's going rapidly. But then again, but if he actually is producing methane, he can also extract the CO2 again from the emissions and then put that into a new cycle.
Yeah.
And then everything keeps turning.
And maybe Gert Jan, you create this PH mainly from wastewater, is that right? And do you think there's enough of it, or is it becoming limiting very rapidly when your PAH technology is ready for the market?
Well, I think we have no shortage of wastewater there. As long as we have more people, if the growing population, everybody produces waste. Yeah,
but we use a lot of plastic as well, I think. So.
Yeah, but there's also a part of recirculation, the reuse of the plastics. So it's not. I don't think it's going to be just one technology. It's just going to be a combination of different kinds of technologies. And you can also wonder, do you need to package everything? So if you go to the Albert Heijn and see the cucumbers and the paprikas and whatever, everything is packaged in plastic. Why? That maybe for now it's good because you have no transmission of your virus into your food and so on.
So we also need to re-educate marketeers.
Yeah, maybe.
Yeah.
Because they have been, I don't know, they've been trained to put things into things to make things beautiful.
So what I hear from you, Gert Jan, that basically the technology is there. Well, what is still keeping us from making PAH on a larger scale?
Competition, price. People need to willing to invest in a factory. And when the price is fluctuating this much and the oil industry has 60, 70 years advantage in optimizing processes. And so we are just starting. Yeah? Our biggest biotechnological processes, the biggest one is the wastewater treatment. Yeah? And it was always made as an end-of-pipe solution. We are cleaning the water so that it can be put into the environment. But only since, well, 10, 15 years, people start thinking, well, wastewater also contains energy and valuable components. So maybe we should change our processes, not to just produce clean water, but to get the valuable components out of it and reuse them. So we invented a lot of processes to remove ammonia from wastewater. Ammonia is a fertilizer, and the ammonia is converted into nitrogen. And then we have a chemical energy consuming process to turn nitrogen back into ammonia to make fertilizers. So why not use the ammonia right away from the wastewater as a fertilizer?
Is that the same as the word struffiet?
Yeah.
Okay.
But legislation prevents us of using material which comes out of wastewater. We don't want to have a direct connection between wastewater and our food.
So any politicians in the public? Then,
I know that the government is working hard to redefine these laws, and it's a tricky route to do so. But it is interesting though that in the animal world, the fertilizer is the automatic outcome of pooing and peeing everywhere we want.
Yeah.
And
these laws also have a function because wastewater contains pathogenic organisms, so you can become ill with wastewater. And so remember a couple of years ago when we had this big E. coli infections on vegetables, and it turned out that these vegetables, AHEC, the AHEC bacteria. And it turned out that the vegetables were originating from Egypt, and Egypt was doing irrigation with partly cleaned wastewater. So there is a need for legislation to do this, but there should also be a law. If you can say that there is no pathogenic organism present anymore, that it should also be allowed to reuse this material.
Yeah, and pathogenic organisms are bacteria which do harm in a chemical perspective.
On a physiological perspective, if you eat AHEC, you have the potential to die.
Because they create havoc in our tummy and our digestion.
In our gut systems, I think AHEC causes hemorrhages, so bleedings internally, and then you will die.
So it eats its way through everything.
Well, yeah. Or no, I think it produces toxin where our cells are not have no defense mechanism against it.
So it's a super degradator.
Yeah.
Yeah, and then,
well, for meat, so for human cells or animal cells.
Yeah, and the interesting thing is that we also have natural processes to get rid of pathogens as well. So nature has also ways to get rid of all the nasty animals. For instance, we just did a lot of measurements on halophyte filters. I don't know the English word for that, but treating water.
I think it's the same.
Halophyte filter. Great. And then you can see that the water that it comes from the wastewater treatment plant still contains E. coli, for instance, and all kind of other nasty pathogens. But when it is filtered by the natural water plants and so on, then you see that you get rid of those. quite fast. So there are ways also to treat, to get rid of these pathogens from wastewater. So maybe
I
we can
think
also.
we have 2 more or 3 topics for next time, Mark.
Yeah, I have a lot of data on that, which are very new and very interesting. So yeah.
Yeah,
Okay,
let's put those separate for next time, Mark, because I definitely want to dive into those also. Are there any ones from the public? There are 5 non-video participants. Do you have any questions for us you want to ask us? Otherwise, we're going to close this podcast down and have the next one in 4 weeks' time. Is there anybody who wants to add or ask something? Nobody is putting their hands up. Okay, then then we're going to close down this one. It was an interesting conversation with you, Gert Jan.
Yeah, I also like the conversation.
And we explored many interesting areas again, and it has created enough new insights for me. And the remarks from Jules were, from his perspective, also very useful.
Yeah.
So it was good to have you here. I hope you enjoyed it.
I enjoyed it very much.
Thank you very much, Gert Jan. Yeah.
Okay, thank you.
I hope you enjoyed this episode of the Master Beautiful Achievements podcast. A special thanks to our sponsors Scope Matters and Orvion for making this series possible. If you like our podcast, subscribe in your favorite podcast app. And if you want to know more, do check out the mastersofbeautifulachievements.com website. If you like this podcast, do not forget to leave us a review on your preferred podcast platform. Thank you for listening and join us next time.